Hello, everyone. Good afternoon or good morning, depending on where you're joining us from today. I wanted to thank you all for joining us for our May webinar and our final webinar before our summer hiatus. We will be picking back up again in the fall, either in August or September with additional MPRINT webinars. But today we are glad to have Dr. Sydney Thomas, Shirley M Tsunoda, and Pieter Dorrestein joining us to give us some information on using untargeted metabolomics to support some of the studies within MPRINT and defining that. So at this point, I will turn this talk over to Dr. Dorrestein. Thank you.
Great, thanks, and also thanks for being here. It's a small group, but so if you have questions, you're always welcome to ask questions throughout the talk, essentially. So what I'm going to do is actually give you a brief introduction of untargeted metabolomics. Shirley is going to highlight how that can be used for molecular pharmacology, and then Sydney is going to highlight how she's been using this in the context of her studies within MPRINT. And so before I go on, I do want to have disclosures. I'm a scientific co-founder of both Ometa Labs and Envita, and then an advisor to Cybell Microbiome. One of the key goals of a lot of the research that we do in a clinical setting is essential to discover biomarkers. And there's lots of different biomarkers. You can have genetic biomarkers, temperature, heart rate, blood pressure, protein. But the focus today will be on small molecule biomarker discovery. And these biomarker discovery efforts are really focused on trying to understand the difference between healthy or unhealthy, monitor health improvements. They can be used for diagnostic purposes, maybe stratifications, or give you mechanistic insight in terms of the disease. And once you have biomarkers in the research setting, some of these will lead to clinical biomarkers. And one of the key technologies that are used to discover biomarkers is mass spectrometry-based metabolomics, which will be the focus of this particular talk. And so there are really two sort of, at the highest level, there are two different types of mass spectrometry-based biomarker discovery strategies. One of those is targeted, where you know exactly what molecules you're going to be looking for, even if it's a panel of molecules. And it's more quantitative and it's more sensitive. So if you know what you're going to be looking for, let's say you want to study the Krebs cycle or specific known bile acids, you would probably want to use targeted analysis. However, the majority of molecules is still uncharacterized in biology and particularly human biology. And so there we have untargeted strategies, and that really has a discovery potential. But as I said, it's less quantitative, but there are some challenges with it. And in the end, most of you know that you may not realize that mass spec is really used in every day, but here are examples of things that your primary care physician might want to monitor: is vitamin D level, creatinine, thyroxine hormone levels. And this is done using targeted strategies, targeted mass spectrometry strategies. And really, this has really revolutionized a lot of the things that we do in a clinical setting. Inborn errors of metabolism, for example, is often monitored, or a lot of the metabolites are monitored using targeted metabolomics approaches or targeted mass spectrometry approaches. Nowadays, microbial identification is also done using targeted mass spec approaches, where you have a library of patterns associated with specific microbes. And so now you can search against those particular patterns and to see if there is a match. And it actually is more widely used than sequencing in the first pass analysis nowadays in terms of screening for what the microbes are. But if you ask, okay, how is untargeted metabolomics used, where you don't know what you're going to be looking at ahead of time, you actually come up sort of empty-handed, and yet it has an enormous potential, because in principle, it should be able to enable you to read the medications as well as their metabolism. you should be able to see all the microbiome-derived molecules, as well as a lot of the, the lifestyle factors that make up human populations. And all untargeted metabolomics actually allows you to detect data on all these different parts of a person, or that makes up a person and their lifestyles. Yet it's really challenging to pull this out, and that is because the data itself is quite complex, and I'm just going to show you how complex the data really is. And so one part is it that we tend to have chromatography separation. So there's a time dimension that is eluded when you separate molecules on a chromatography column. And then we have a detection component to it, and their relative intensity is usually associated with the amount of the molecule that is present. But different molecules have different ionization efficiencies, and thus some of them are harder to detect than others. Then you also have the ability to isolate any of these molecules, or really ions of molecules, so charged versions of the molecules. You can isolate those very quickly inside a mass spectrometry and break them apart. So now we have these patterns that basically are connected to the structure of the molecule. And so you can think of it as sort of a unique barcode that you have per molecule. And we can do this very rapidly. So in a given experiment, we may get 2,000 to 5,000 of these MS2 signatures that we can then detect. And really, this is the speed by which it happens. So here's the speed with MH1, usually every half a second or every second. So these are all the intact ions. And then the fragmentation patterns are collected at this speed. And so you can start to see how much data we're going to collect. But the other thing that you should notice is that that data is quite noisy. And so we need to be able to deconvolute that noisy data and be able to assign molecules to it. And that's really one of the challenges. And then one molecule might have many different ion forms. So here we have a representative bile acid. And they are represented by multiple different adducts, so protonated, ammonia, sodium. Maybe there are even others. Then there are in-source fragments where the molecule breaks up before it's detected by the mass spec instrument. And then you have multimers formations of these ions. And so one molecule can be represented by a large number of different ion forms. And so here's an example of this bile acid. These are all the same bile acid, and it's represented by so many different ion forms. And so we need to learn how to deal with it. And then there's the issue of the reference library. These are not comprehensive. So what we do is we usually search these MSMS spectra or MS2 spectra against libraries of known molecules, and that's how we provide annotations. And so in 2016, we had 2% of these annotated. And then the final challenge that exists is that molecules undergo metabolism. So here we're just looking at diphenhydramine. And so you can see a glucuronylated version, oxidized version, and even an acylated version over here. And many molecules undergo metabolism in this similar type of fashion. And so we need to be able to recognize that even if the libraries are not available. And this goes back to that shoe picture that I showed you. Basically, there were two shoe salesmen. They were opening up new markets. They arrived in the new market, and one salesperson sent a telegram to his boss and said, well, situation is really hopeless. Nobody's wearing shoes here. The other person, however, said, oh, well, this is a great opportunity. Nobody has shoes yet. There's a huge market for it. And so we took that positive sort of view and said, okay, this is an enormous opportunity. So we've been building tools for the community to analyze untargeted metabolomics data. And that is now accessed 450,000 times a month. And I'm just going to highlight some of the things that we have been able to develop. The first thing that we've developed, and that's really an important foundation, is where we can take these MS2 ions or MS2 spectra. And first we merge all identical spectra, and then we look for similarity in the same way that you do sequence similarity for looking for mutations and such. And so this allows you to build a network of these spectra. And then if you have one known molecule, such as caffeine, it may be linked to the other node by a 14 Dalton, for example, theobromine or paroxetine. And And that's because they're missing one of the methyl groups here. And so now you can begin to propagate some of these annotations. And this is really what it allows you to do. It allows you to build a knowledge graph. And so here we have a knowledge graph for feces, and we can see the number of annotated spectra and the number of unannotated spectra. And in this case, it's about 5% of the molecules are annotated and the rest still remains unannotated. But I want to show you how we can use the ability to network this information in the context of doing a food readout from clinical samples. And so the idea that we had is, well, all the molecules that exist in your body essentially come from ingestion, either food or beverages, and maybe some absorption through your skin, but the majority comes from diet. And so the idea that we had is that, what if we collect data on these clinical samples, we should be able to get a dietary pattern, or at least proportional dietary pattern in relationship to all the other patients that are part of the cohort. And the way this is done is that we create data on reference data and clinical samples, and we create this knowledge graph using molecular networking. And using this knowledge graph, we can now translate this into a food ontology, clinical samples, and relative abundance associated with these particular food specimens that we have. And so I'm going to show you how we use this in the context of IBD. And so here we did a PCA on the proportional diet readouts that we were able to obtain from this particular data. And so in circles, you'll see Crohn's disease patients, in triangles, healthy controls, squares, ulcerative colitis. And then we provide the proportion of the fleshy fruit. And so here you can see that the people on the left have more fleshy fruit associated with them. People on the left have more vegetable associated with them, and the people on the right have more meat products associated with them, and on the top, fermented grapes. And now what we can do is we can use low-level machine learning strategies. Usually simple regression analysis is sufficient to associate clinical metadata to these particular dietary patterns. And so if we look at the association with disease, so ulcerative colitis, Crohn's disease, or healthy controls, there doesn't appear to be diet changes in those three different disease states. No dietary patterns associated with age. So in other words, young people don't eat different diets than older individuals, at least in this cohort. And then sex, males and females in general eat similar type of diets. But now if we look at microbial diversity, or phase PD, now you can see that there's a higher diversity associated with the microbial communities in relationship to the dietary patterns that are observed. And it's along the PC1 axis. And so the more fruits and vegetables you consume, the higher the microbial diversity there is. And so that association has been documented in the literature as well. So it's nice to be able to recuperate that in this particular example. And then we have disease activity. So this is based on endoscopic scoring. Now you see that disease severity is much worse the more sort of red meat is consumed. And then there's less disease activity, at least according to the endoscopic score, the more it focuses towards fermented grapes. So it's sort of the first time that we're starting to be able to get empirical dietary pattern readout and associate this with clinical phenotypes. And so in the future, we hope to do clinical trials with some of these observations. And so I've shown you really briefly food, that we can do food readouts, and we're moving towards doing microbial readouts. So we've started to organize the 8,000 LCMS-MS files of cultured microbes in the same way that we did for the food. And we have organized these according to specific patterns. So if you look for the MS2 spectrum of Yersinia bactin, we find that it's associated with E. coli and Klebsiella. Yersinia pestis is not in there, otherwise it probably would have shown up as well. And then here's a new bile acids that we have been uncovering that are produced by microbes. So instead of your typical glycine or taurine conjugated bile acids, we've started to find a lot of different microbial bile acids that are conjugated with different amino acids, even non-proteinogenic amino acids. And we basically now see that they're produced by a lot of different microbes. So the microbes are really responsible for creating these different bile acids. I said there were only 2% annotated in 2016 because of the infrastructure that we've built and other people have been contributing. We're starting to get a lot more annotations, but it is sample type dependent, because if we look at the human data, you can start to see that blood has about 20% of the data annotated, brain about 25%, CF fluid more than 30%, feces average is 10%, although some of them we see up to 60%. So there's huge diversity in terms of ability to annotate fecal material. Milk, it's average 15%, et cetera, et cetera. So you can see that the amount of annotation will be dependent on the sample type, and often it's related to how much it's been studied. And then finally, the things that I highlighted are really due to an amazing team that I have. You will hear from Sydney in a second. And before I answer questions, we'll do that at the end. I will pass the baton on to Shirley.
Great, thanks, Peter, for setting that up. So I'm going to now take all that information that Peter just gave you on the infrastructure that he has built, and I've been fortunate enough to be able to take advantage of that infrastructure and start to use the tools that Peter and his team have built in the context of clinical studies. So I'm going to walk you through a few clinical studies that we've performed utilizing untargeted metabolomics and sort of hopefully illustrate for you kind of the power of untargeted metabolomics. And, most of you may know, if you're in the pediatric maternal space, that we have difficulty monitoring drugs, right? So it's really hard to get blood from babies. It's hard to get blood from pregnant women. So is there a non-invasive way to measure drug levels, drug detection? And so we piloted a study, and this is together with Alan Yarmouche, who was a postdoc in the lab, because I see transplant patients and we monitor our immunosuppressive drugs daily sometimes when they're in the hospital, and we have to sample their blood. So wouldn't it be amazing if we could non-invasively monitor their drug levels? And previous work in the Dorrestein lab had shown that drugs actually do show up when you just non-invasively swab the skin. And so what we did was we took this paradigm of, it's kind of basically like a Q-tip that's been soaked in an ethanol water solution. And we just rubbed this in a circular motion for about 10 seconds with moderate pressure. And we did this in various parts of the skin to see if there were differences, whether we saw the forehead, the nasolabial folds, fronts and backs of the hands. And we did this in kidney transplant patients. And unfortunately, we did not detect immunosuppressive drugs, but we did detect 5 other orally administered agents. So many of these patients are taking, 20 to 30 different drugs. So we knew which drugs they were taking, and we unfortunately didn't see their main immunosuppressive agents, but we did see citalopram, an antidepressive agent, trimethoprim, an antibiotic, diphenhydramine, common antihistamine, sulfamethoxazole, also an antibiotic. And then interestingly, we saw the N-acetyl metabolite of sulfamethoxazole, but only in the axillary regions of the patients, of a couple of patients. So you can see we saw kind of differences depending on the different parts of the skin that we sampled. And so that study kind of spurred us to do a more prospective study to really show, what's going on here? Obviously we're seeing drugs not just topically applied, but these are drugs that patients ingest, And then somehow these drugs are making it to the surface of the skin. And this pathway and these mechanisms are not very well characterized. And so we decided to just do a pretty simple study in healthy volunteers, since we'd seen diphenhydramine and it's, over the counter available, it's safe to take. And we just gave 10 healthy volunteers a 50 milligram dose of diphenhydramine. And we drew their blood and we sampled their skin. And this was, a lot of the work was done by Morgan in the lab, sorry. And what we found was that, and we did targeted. So, Peter talked about targeted versus untargeted metabolomics. So Jeremiah Momper created an assay to look at diphenhydramine parent compound. So we have a nice, pretty characteristic concentration time curve of the parent drug diphenhydramine. And then using untargeted metabolomics, we will able to also look at parent diphenhydramine in the blood and in 3 different areas of the skin. We looked at the upper back, the forehead, and the forearm. And as you can see, using untargeted metabolomics, we were also able to see some of the primary metabolites of diphenhydramine. So we didn't have standards of metabolites to be able to see those in the targeted assay, but because untargeted metabolomics can see, virtually everything, as long as we have these annotated in our databases, and then we can infer others based on the molecular networking that Peter outlined. So as you can see, we saw these nice kind of, and I hesitate to call these concentration time curves, but this is over time. And then these are relative peak areas of both diphenhydramine and 2 major metabolites. And you can see that overall, all of the skin, all of the appearance and disappearance of the skin lagged that of the blood by several hours. So, the drugs or the metabolites appeared in the blood, peaked, and then several hours later, we saw it show up in the skin. And then interestingly, we also saw the glucuronide, the secondary metabolite of diphenhydramine, but only in the plasma. And same with this novel metabolite, that we identified in the context of identifying the glucuronide, but this was also only seen in the plasma, but not in the skin. And as you can, this is just showing you the data in a little different way with all of the skin and, or all of the parent and metabolite data by matrix. So this is plasma. The forearm skin, as you can see, was not that great at picking up many of our compounds, but the forehead skin and the upper back skin was much better at picking up several of these diphenhydramine parent and metabolites. And then this is just to illustrate how we can use untargeted metabolomics to identify previously unknown metabolites. So this was while we were looking for the glucuronide, we found a separate peak that we annotated as diphenhydramine glucose, which was distinct from that of the glucuronide. And Emily in the lab, who's a synthetic chemist, was able to actually synthesize that N-glucose and show that the synthesized standard was pretty much superimposable to the one that we saw in our samples. And again, it was only seen in the blood, but not in the skin. So this brought us to the question of, okay, so which drugs do we see in the skin and which ones don't we? We don't see immunosuppression. So are there certain characteristics of drugs that can help us to predict which drugs we will see on the skin? Because presumably there's a pathway that goes from the blood to the surface of the skin. And what are the physicochemical properties of drugs that dictate which drugs are going to make it to the skin? So Wout in the lab devised a random forest classifier to, and we took skin samples from all of the various studies that the Dorrestein labs collected in the publicly available database. And we had some negative examples of drugs that we knew patients were taking from that kidney transplant study that I showed you before. And those drugs were not seen, many of those drugs were not seen on the skin. And he trained this random forest classifier to try to make a prediction on which drugs would make it to the skin and which ones would not, based on physicochemical properties. So he used the Mordred classifier, which classifies various molecular descriptors of drugs, and then he used the SHAP feature importance to try to characterize which were the most important features, molecular features of drugs that would help us predict which will make it onto the skin and not. And so most of and these are, I know these kind of don't make a whole lot of sense, they're just abbreviations, but most of these physicochemical properties have to do with electrical topology and bond energy and van der Waals forces of drugs. We tried to collect pharmacokinetic information, such as, volume of distribution, protein binding, things like that, half-life. But unfortunately for the drugs that we had available, we did not have enough comprehensive pharmacokinetic information for all the drugs. So unfortunately, we couldn't utilize those types of characteristics to make predictions. We also kind of roughly looked at molecular weight, because if you can just kind of think of the size of the drug, maybe would help predict whether or not the drug's going to make it to the skin. And as you can see, in general, larger compounds don't make it to the skin. And then what he did was he also was able to show that we had some experimental data. Remember that diphenhydramine study that Morgan did? So sorry. And he ran the diphenhydramine through the model and showed that, yes, in fact, diphenhydramine is likely to be seen on the skin based on his model. Whereas the hexose, remember that metabolite that Emily synthesized and that we saw only in the plasma but not in the skin, would not be likely to make it on the skin. Citalopram, also that we saw in our kidney transplant study, is likely to make it on the skin, but not tacrolimus. And as you can see, the blue, tacrolimus very strongly likely not able to make it on the skin. So this sort of kind of confirmed that our random forest classifier was pretty reliable in matching our experimental data. And then Wout took over 2,500 FDA approved drugs and ran those through the classifier along with many of their metabolites. And just to kind of see, could we classify drugs based on their therapeutic class to determine whether or not they're going to make it on the skin? And in a way we sort of can, but you can see there's also a lot of variability, and that's likely because there's a lot of physicochemical differences within any therapeutic class. But in general, we saw that nervous system related drugs, pain medications, for example, might be more likely to be able to be seen on the skin as opposed to immunosuppressive drugs, as we saw experimentally with tacrolimus. Now, we can also use untargeted and targeted metabolomics as tools for pharmacomicrobiomics. So pharmacomicrobiomics is that intersection of microbes, our gut microbiome, intersecting with drugs, and in particular, absorption, distribution, metabolism, and excretion of drugs. So just to remind you, the pathway of an orally administered drug goes through the gut and encounters not only drug metabolizing enzymes and transporters, but also notably the gut microbiome throughout the length of the GI tract. So that drug is going to lose, part of that bioavailability is going to be lost due to many factors, including enzyme, enzymatic metabolism, transporters that work together with the enzymes, gut microbes that work directly by metabolizing drugs directly, but we also think they interfere with our host drug metabolizing machinery to impact the fate of the drug before it makes it to the liver. I'm so sorry. Before it makes it to the liver, where it also will find cytochrome P450 enzymes and transporters before the drug makes it to the systemic circulation. So we did a study, and this was again Alan Yarmouche, postdoc in the lab, where we took healthy volunteers and we gave them a cocktail of caffeine, dextromethorphan, omeprazole, and midazolam. And for those of you in drug metabolism, you'll recognize this as the Cooperstown cocktail. And these are drugs that we're using as probes for the cytochrome P450 enzyme system. So caffeine is a probe for 1A2, dextromethorphan, a probe for 2D6, omeprazole, a probe for 2C19, and midazolam, a probe for 3A4. So we sampled their blood, urine, and feces. We took a fecal sample before we gave any drugs as well to get a baseline of their microbiome. And then we gave this cocktail and did a pharmacokinetic study, sampled their urine and their blood. And then we gave them cefprozil, a second-generation cephalosporin, in therapeutic doses, 500 twice daily, sent them home, and they collected feces every day. And then we replicated this pharmacokinetic study by giving them this cocktail of drugs. We also sampled feces several days out because we wanted to see what the recovery was post-antibiotic. And what we found was that the antibiotic, and the antibiotic is not known to impact any cytochromes or have very many drug-drug interactions. It's mainly excreted unchanged in the urine, although I'm going to show you a little bit of data that shows that it is seen in the feces. And we found that the 1A2, 2C19, and 3A4 activities were all decreased after administration of cefprozil. And we don't think that's because of any kind of inhibitory effect or on the enzymes by the antibiotic, but we think that is because, we're hypothesizing it's because of the impact on the microbiome. And there's animal data to suggest that this is the case, that the microbiome that possibly metabolites of microbes can interact with our replicating machinery of the cytochrome P450s and can modify the activity of those enzymes. And we saw that alpha diversity, which we would expect with antibiotics, was decreased over the course of the antibiotic and then recovered to baseline levels at the end of the 30 days. And we did see characteristic decreases in several genuses and families of organisms. We did 16S rRNA we did not do metagenomic sequencing in this study. And then we can also see Alan did a molecular network where, like what Peter was explaining to you, where we could make some relationships to our known compounds and see these known metabolites of dextromethorphan, midazolam, and omeprazole. So we saw both phase 1 and phase 2 metabolites in these samples. And then finally, we can use untargeted metabolomics and molecular networking to identify previously unknown metabolites. So Alan took omeprazole. He saw that there were some related nodes that were unannotated. So these purple and the lighter white colors are, these are all spectra that are related to omeprazole, but don't have a clear annotation in the libraries. And so he found this one node here correlating to omeprazole sulfide, which is a known metabolite of omeprazole that is modified by gut microbes. But then there was this other related node, differed by about 16 from the omeprazole sulfide, that we are postulating is possibly a 5-hydroxy omeprazole sulfide. And this 5-hydroxy is a common way that cytochrome 2C19 modifies omeprazole. So we postulated that this metabolite, 5-hydroxy omeprazole sulfide, was both a gut microbially modified and a host human 2C19 modified metabolite. And we wouldn't have been able to know any of this if it hadn't been for untargeted metabolomics, because this is not known a priori. Similarly, Alan found in our cefprozil samples, so this is the same study where, remember, we gave cefprozil, and we saw cefprozil both in the urine and in the feces of patients. Cefprozil, there's not a lot known about its metabolism. There are not any known hepatic metabolites, although it says that, in the literature, it says that hepatic metabolism occurs. And we saw that there was a putative metabolite that we're calling a reduced cefprozil that also occurred in the feces during the period of time that the antibiotic was taken, during the time that cefprozil was taken, those days 2 through 8. So again, just another example of finding metabolites that are not previously known in the literature based on connecting the dots of their chemical relatedness to known compounds. So just to summarize, untargeted metabolomics in the context of clinical pharmacology can be a powerful tool to uncover previously unknown pathways of metabolism. We have been able to kind of infer origins of drug metabolites, kind of differentiate between host and microbe. We've seen that it can be used as a tool for non-invasive monitoring, which is a huge advantage, particularly in the maternal pediatric population. And then we've also used it to investigate drugs and drug metabolites in the context of the entire chemical space, because we see microbes, food, environmental chemicals, and all of their metabolites in this chemical space of metabolomics. And I'd just like to acknowledge all the people who've done a lot of this work, and particularly Peter and Rob Knight, who've provided amazing infrastructure for being able to interrogate a lot of these questions in the clinical space. And now I will hand it off to Sydney.
Thanks, Shirley. All right. So I'm Sydney Thomas. I'm a postdoc in the Dorrestein lab, and I'm going to give a very brief vignette about how we can use all of these really interesting techniques that Peter and Shirley have described to actually improve infant health in MPRINT. So this particular project focuses on human milk, which, as you know, is a very crucial part of normal, healthy infant development. But despite the fact that we know that breastfeeding is associated with all sorts of positive infant health outcomes, we still don't really understand what exactly in milk is responsible for those effects. However, to be able to answer that question, we first have to figure out what's in milk in the first place. And so to start to ask that question, I pulled untargeted metabolomics data from about 1,000 human milk samples that are publicly available on the GNPS database, which is run by our lab. An analysis of those samples produced around 21,000 spectra. So this corresponds to tens of thousands of unique molecules that show up in human milk. And we were able to annotate about 5,000 of them. So this is actually a pretty good annotation rate, as Peter was saying. However, this still leaves this vast metabolic gray area where we know that these compounds exist, but we don't know what they are. Thankfully, molecular networking allows us to kind of wade into that metabolic gray area and figure out the chemical identities of these unknown compounds. So, for example, this is a molecular network of sulfa antibiotics in human milk. So in this particular molecular network, we actually know the identities of some of these compounds. In particular, the green nodes or circles in the middle of the molecular network here correspond to the antibiotic sulfamethazine. Now, that means that even though we don't necessarily know the chemical identities of all of the other compounds in this network, we do know that they're structurally related to sulfamethazine. And so we can dive into that raw mass spec data and actually get a pretty good educated guess of the chemical identity and structure of these compounds. So this is a little bit of what that looks like. On the left, you can see what are called mirror plots. So the black lines on the top of the mirror plots correspond to the spectra of our known chemical, in this case, sulfamethazine. So this is what we know sulfamethazine looks like when we put it on the mass spec. And below the green and gray lines correspond to our unknown metabolite of interest. And so here you can see there are several green lines, which correspond directly to the black lines above them. And that indicates where the structures are identical between the two compounds. And then the gray lines that don't match indicate where the structures differ. And so using this information and some chemical know-how, you can actually get a pretty good educated guess at the structures and names of these unannotated compounds. And so the real power of this method is that instead of just looking for that one antibiotic that you know should be in the sample because you know you gave it to the patient, instead what you're seeing is the entire constellation of antibiotics and antibiotic metabolites that actually exist in milk, many of which are novel. They haven't been reported in the literature, and we don't have any data on their activity. So this is really exciting, but this is easily not the only medication that we can see in milk. For example, here are some representative molecular networks of some antidepressants, amitriptyline and bupropion. You also see many molecules that aren't necessarily related to medications. For example, the molecular network on the bottom right here in green corresponds to the Pseudomonas quinolone signal. So this is a molecule produced by Pseudomonas bacteria to detect how many other bacteria are in its environment. And so this highlights the fact that milk is definitely not sterile. There are bacteria in milk and they are affecting milk metabolism. So while this is all very great, it really highlights the diversity of compounds in milk. Just because we can detect a compound in milk does not mean that that compound will then be transferred to the infant. And so to get an idea of what's happening here, we turned to a preliminary data set that contained 40 parent-infant dyads where we had untargeted metabolomics data from the parent's milk, as well as the infant's oral, skin, and stool samples. So these are generally just like cheek swab samples or stool samples, for example. And on the left hand here, you have some representative molecular networks for some antibiotics, as well as some unknown metabolites that are very likely of bacterial origin. And the colors within those circles there correspond to which sample type we saw that particular metabolite in. And then on the right here, we have the frequency with which we saw different drugs in the parent-infant paired samples. So you can see in the case of azithromycin and discladenosid azithromycin, the antibiotics, in the vast majority of cases, if you see that antibiotic in the parent's milk in yellow, you will then see that antibiotic in the paired infant stool in blue. So that highly suggests that this particular antibiotic is being transferred from parent to infant via milk. It's traversing the infant's entire intestinal tract and then being excreted in the infant's stool. This is not the case for every single medication. As you can see with the case of acetaminophen, kind of in the middle there, there are several instances where we saw acetaminophen in an infant's oral sample where we didn't see it in the parent's milk. And so that would strongly suggest that that infant was given acetaminophen directly instead of having that drug be transferred from parent to infant via milk. So this is something we're planning to greatly expand our data on with some of our MPRINT clinical studies. And we're really excited to see which drugs are more likely to be transferred and how many drugs we can actually see in those larger data sets. So this is all really great. We have a lot of metabolites. Many of them can be transferred from parent to infant. But what we really want to do in the end is use this information to improve infant health. And so one of the ways we've been doing that is by looking at a data set that it contains 550 human milk samples, where we have several high throughput data methods collected. So we have 16S sequencing data for the microbiome in the parent's milk, untargeted milk metabolomics, and a high throughput method to measure human milk oligosaccharides, which are sugars that are very specific to milk. And what's really powerful about this data set is not only do we have, this massive amount of data on, different microbiome and metabolome stuff, but we also have a lot of really interesting metadata concerning medical conditions from the parents and all that kind of stuff. And so what we can do here is actually look at how each of these data types affects medical outcomes. So one of the first ways we did that was to look at the diversity of each of these data types and how they change with different medical conditions. I'm only going to show the microbiome and HMO data in this particular figure because it's already messy enough as it is. I know this is a lot. So we're going to focus on a couple of different types of conditions. And you can see that, for example, when the parent uses opioids or has a preterm birth, what we see here is that their microbiome diversity goes down while their HMO diversity goes up. And so that was really interesting to us. And we wanted to see if any specific HMOs were responsible for this effect. And so we correlated every single HMO that we measured to microbiome diversity. And you can see here that many HMOs are either positively or negatively associated to microbiome diversity. But what was really interesting to us was that 3 HMOs in particular, 6SL, DSLNH, and LSTC, were not only very well correlated to low microbiome diversity, but they were also significantly upregulated under several medical conditions. For example, having a C-section or preterm birth. And so we presented this data to our collaborators who are HMO experts, and they said, well, obviously all of these HMOs are acting in a similar manner because they actually share similar structures and are created by the same enzyme, ST6-gal1. So if we can look at all 3 of these HMOs together and their concentrations and stratify our samples by parents whose concentrations of these 3 HMOs are in the upper quantile versus the lower quantile, what we see is that parents in the upper quantile were 2 times more likely to have had a C-section, twice as likely to use SFRIs, and 5 times more likely to have had a preterm birth. And what's really interesting about this is not only do these HMOs seem to be associated with, kind of more poor outcomes at birth, but they seem to have knock-on effects on infant development over the first 3 years of life. So you can see here, children of parents whose HMO values are in the upper quantile have more issues with externalization of emotions at 12 months and internalization of emotions at 30 months based on the infant-toddler social-emotional assessment. We also see that they have lower percent of phrases understood at 24 months using the McArthur-Bates Communicative Development Index. And then in panel C, you can see that the actual concentrations of these 3 HMOs together are correlated, significantly correlated to dysregulation of negative emotions at 36 months for these children. So this is an incredibly interesting phenotype. And what we really want to know is why in the world this is happening. And so one of the first things we did was to see if there are any specific bacteria that might be associated with these HMOs. And so to do this, I collaborated with another postdoc in our lab, Dr. Simone Zuffa, to correlate our microbiome data to our HMO data. And what he found was that there are many microbial genera that are associated with 1 to 2 to 3 of these HMOs together. Here, HMOs are shown in the triangles and the different microbial genera are in the circles. And you can see that in the center here between those 3 HMOs, there's a bunch of different microbial genera that are associated with all 3 HMOs in the same direction. And so that strongly suggests that these HMOs are changing the microbiome of the milk. But we also need to know what happens when those microbes are changing. Why is that important? And so one of our next steps is to really integrate our untargeted metabolomics data to figure out what are the metabolic knock-on effects of having high concentrations of these 3 HMOs. So I actually just took this data off mass spec a couple of weeks ago, so I don't have any beautiful figures yet, but we are seeing some differences, especially in some phosphatidylcholine molecules between, for example, term and preterm birth. And we're hoping to get a lot more data from this in the future. But in general, what we've found so far is that human milk is incredibly metabolically diverse, and that this composition can be predictive of things like preterm birth and infant development in ways that are very surprising. And that integration of this multi-omics type of data, untargeted metabolomics, microbiome sequencing, HMOs, can give us new insight into how milk composition affects parent and infant health that we may have never gotten if we had just looked at each data type separately. So I would like to thank all of the people that helped with this data and analysis. It really takes a village to analyze all of these samples. And I'm sure any of us would be happy to answer any of your questions. Thanks.
Great, thank you to all of our speakers today. Are there any questions from the audience at this time? If you have anything, you can type it in the question and answer box or raise your hand and I can allow you to speak. Anything yet? I do have a couple of questions. Shirley, on your discussion of the drugs that get out onto the human skin, do you have any explanation for why some appear at different places and why the variability in the locations?
Yes, that's a great question. So that's one of the reasons why we sampled at different parts of the skin. So that one example I gave you with the N-acetyl metabolite of sulfamethoxazole being a more polar molecule that we think only showed up in the axillary areas, which is a more aqueous environment, sweat, etc., as opposed to the forehead or other areas of the face that are have more sebaceous glands, a little more oily. And so we do think that not, there is going to be differences depending on the different parts of the skin that we're assessing based on the chemical of the drug, and the properties of the drug, whether it's hydrophilic, hydrophobic is a kind of a simple way of thinking about it, but there are probably more, other things like transporters and, other characteristics of the drugs that dictate where it's going to show up on the skin. So we're still at the very early stages of understanding these pathways. And we also, with our method, we think we're sampling skin cells because we're lightly rubbing the surface of the skin. There are other investigators who think that they are looking at sweat. So it's a little unclear. Again, like, are we pulling off a little bit of sweat in combination with some of these skin cells? And that might also cause some variability in which types of molecules that we see. So great question.
Thanks. Have you looked at things like the transporters and whether the cellular uptake is correlated with exposure in skin at all?
Yeah, no, I mean,
data.
Other than question, which transporters are here, what are they transporting? I mean, I don't know which transporters are on what spots of the skin and if any of these drugs are and unfortunately, we just didn't have, again, the comprehensive database of the drugs that we saw on the skin and which transporters transport these drugs, and then again, whether or not these even exist on the skin. So this mechanism and the pathway of how these drugs get to the surface of the skin is not known.
Sydney, a question for you on the panel where you showed that there's a variability between maternal and the pediatric exposure or expression of these metabolites in the oral cavity and so forth. Do you know if, do you have information on the drug intake by the mothers and infants in that study and whether you could confirm that, perhaps they gave the baby acetaminophen and didn't take it themselves?
Right. So for this particular study, since this was publicly available data that we did not collect ourselves. We don't have that information. That is one lovely thing about untargeted metabolomics, is that we don't need information like that to actually detect the drugs. But it also leaves us without ways to confirm. But that is one thing that we'll know in the new clinical studies that we're doing with MPRINT, is actually what drugs the patients were taking. And so that will be very nice to get.
Yeah, I also thought it was interesting that sometimes you would see it in the oral cavity, but not in the feces or vice versa there. And, you know, the explanation of that, you know, whether it's a PK, you know, when baby fed and
right,
that
exactly.
aspect would be interesting to look at as well.
Yeah, and unfortunately also in this particular study, we don't know when each site was sampled. They could have been sampled at different times. And so there's a very large possibility that, you know, the milk was sampled later and so we didn't see it there, etc. So there's a lot of timing that goes into this kind of thing. So.
I think, you know, another aspect I'm interested in too, and I guess probably any of you can answer this, would be how quantitative is the data that we see? I know there was some in Shirley's talk showing, you know, kind of a PK curve from the skin, but it didn't quite match. And then, you know, are we able to do any quantitation with the untargeted approach, or utilize a targeted approach potentially to be able to?
Yeah, Sarah, so I'll answer and then maybe Peter can follow up, but it is, you know, it's relative quantities, right? So we can certainly make conclusions based on their relative quantities to each other. But we actually had a comment about this, somebody at the University of Vienna who wanted to quantitate our untargeted levels, because yeah, you know, as a pharmacokineticist, like you always want to try to create a, you know, calculate an AUC. But unfortunately, the methodology for how the mass spec instrument was set up for the untargeted approach does not lend itself to correlating with our targeted approach. So that was one of the reasons why we also did targeted metabolomics and created an assay with diphenhydramine, because we wanted to just make sure that our population was, you know, showing the PK characteristics that were, you know, characteristic of diphenhydramine, and we didn't have some, you know, weird, you know, blood absorption or, you know, absorption of the drug. So it's our current methodology for untargeted is not quantifiable, but, you know, in thinking about how to move this forward, we could certainly make it quantifiable by using the proper, you know, mass spec recipe for quantitating. So that's certainly something we could do in the future, but we couldn't do that with this current approach.
Yeah, I think that's largely correct. So we can begin to think about making it more quantitative. There are some challenges with it. How hard do you swab, etc. But if you looked at the targeted data in relationship, that was quantitative in relationship to the relative amount that was present for the compound, in the case of diphenhydramine, you can actually see that the curve actually follows perfectly. And so that's not the issue. So what needs to happen essentially, if you know what compounds you're going to be looking at, you could add a C13 labeled internal standard when you do your mass spec, and then you have better quantitative capabilities, but then and still have the discovery component to it. So for example, the new glucose addition to diphenhydramine, which was a new metabolite that we were able to observe. If you don't have a standard, you can't do the quantification, but you can still do the relative quantification of that particular molecule. And ideally, you have a C13 labeled standard, because different compounds have different ionization efficiencies. There's sometimes some matrix interference that can skew the signal a little bit. But generally what I've seen is that limits to about 20% of the total value. And so matrix effects are real in terms of what we talk about in metabolomics matrix effects. But they are not as large as what most people really describe. And so really, if we really want to push this quantitative analysis, is you need to have a C13 labeled standard. or N15 labeled standard.
I think that we're out of time for today. Thank you all for an excellent presentation. And I know I certainly have learned a lot about metabolomics and the applications we can use here for it. So hopefully we can encourage others to utilize the technique as well. So thank you. And we also invite everyone to join us for our MPRINT annual meeting, which will be held virtually June 13th to 14th. So watch for information by email on our website or through one of our social media platforms. So thank you. Have a good day.
Thank you.